This protocol can be used to study the mechanisms governing the invasion of cancer cells into the extracellular matrix in real time. The main advantage of this technique is that it uses a simple spheroid imaging device, which makes the spheroid invasion assay easier to set up and improves its efficiency and cost.
While we demonstrate using the spheroid invasion assay to model mammary carcinoma, this assay is an excellent model for the invasion of any solid tumor into the surrounding healthy tissue.
After 3D printing of the spacer, weigh out a 10:1 ratio of base polymer to crosslinker in a plastic cup, and use a disposable pipette to thoroughly mix the resulting PDMS solution in the cup. Place the cup into a vacuum chamber, and quickly release the vacuum pressure to remove any air trapped at the surface of the mixture, and to dissipate the remaining air bubbles. Incubate the 3D printed spacer at 100 degrees Celsius for 5 minutes to increase its flexibility, and clean 2 glass plates with 100% isopropanol. Place the spacer so that it is flush between the 2 cleaned plates, and place 2 large binder clips on the bottom edge and 1 onto the top corner to seal the mold onto the outside edges of the plates. Inspect the top part of the mold to ensure that the spacer is flush with the glass plates, and use a disposable pipette with approximately 2 centimeters cut from the tip to slowly but continuously add the PDMS mixture to the top left corner of the mold. When the entire mold has been filled, place the mold into the vacuum chamber.